everyone, this is actually the lecture for Thursday, August 20th. I'm posting it a little bit early so that hopefully you can at least look over the slides before Thursday's lab. So during week 1, the first lab was on basic aseptic technique and microscopy. The second lab is really about staining. So I did post the slides for the third lecture, which is Tuesday of week 2. It's basically a review of just basic biological chemistry or biochemistry. So thinking about it through a microbiological context. I know that that's kind of the start to, again, Bio 5, anatomy, physio, and now micro. So it might be stuff that you're already very familiar with. That's why I think it's more important to record this lecture first, especially because some people will actually be staining on Thursday of week 1. So again, with this kind of combination of synchronous and asynchronous, things are going to feel a little bit chaotic just because we're doing the best we can to make sure some of you are able to learn those techniques hands-on and the rest of you are able to at least observe it. So one other thing that I wanted to add along that note is if you join the session live, the Zoom format is going to allow you to really visually see the classroom. What I'm going to do tomorrow when I come in for the second lab is set it up so that the slides are visible for much of the recording, but then I'm going to close the slides so that the Zoom captures in a larger screen what's happening in the classroom. That way, as I model negative staining and have people practice gram staining, if you are watching the recording at a later time, you'll be able to see it as effectively as people who join the live Zoom session. So again, we're just kind of troubleshooting, figuring out how to really capture the full experience. I also think that there might have been a situation where maybe some of the stuff didn't get picked up by the mic when I was in the other room, but I'll have to watch the recording just to double check. So in terms of the actual flow of the lecture, on the first day of class, we kind of talked about the overall diversity of life and different things that we consider microbes. The second lecture was about microscopy, kind of the physical and light aspect of it. The next lecture, which again, I haven't recorded it yet, but I have posted the slides, was a review of biochemistry, the chemical level of organization, specifically in microbes. And this lecture is really looking at the specialized anatomy and chemistry of cells to understand the chemical interactions that happen between those structures and dyes so that we're able to look at them under a microscope. So everything was kind of building up and coming back together. So the learning objectives here are to take those ideas that you kind of explored in the second and third lecture on microscopy and biochemistry and integrate that with functional anatomy. So the way that this lecture is set up, I will kind of talk about a general technique or a structure in the cell, and then the staining that we do to visualize it. So it's kind of broken up roughly according to functional anatomy and then the technique that we used to explore that particular functional anatomy. We'll also think kind of broadly about these steps of slide preparation. That was what you did in the main microbiology lab room on the first lab. So when you were practicing aseptic technique, flaming your loop, putting it into the tube, transferring that onto a slide, that is the first part of slide preparation. And then we kind of talked about how it would look to both dry that out and heat fix it on those slide warmers. We'll think specifically about simple and negative stains, differential stains, and structural stains. So thinking again about simple, gram, negative, just to be clear there, that's gram stain and negative stain, two separate things. Endospore stains, acid-fast stains, and capsular stains. And then we'll also be practicing identifying particular organisms based on their functional anatomy and how that is visualized through staining. I know that for some of you, if you haven't taken one of my classes before, it can seem like I'm just kind of reviewing things very slowly. Everything I do when I design my own slides is extremely intentional. So I am really trying to make sure that you have all the puzzle pieces in place so that when you learn the new material, you're reminded of the pieces you've already put together, what you already know. So this kind of study guide approach and then reminder of that foundational information is really important for giving meaning to the rest of the lecture. So I hope you're not skipping these parts. Sometimes I'll show you slides that I've already shown you before. For example, this is from that lecture that I have not recorded yet on biochemistry, but we explore different types of antibiotics. So thinking about the structure of the bacterial cell wall, and sometimes we're able to target that cell wall structure. Sometimes bacteria are able to use special proteins called enzymes to fight the tools that we have. So again, really thinking about that structure-function relationship. In that lecture, we also talked about how the cell walls and membranes of many different categories of bacteria consist really of basic chemical groups. So things like fats, proteins, carbohydrates, and then also deeper down into the cell, nucleic acids. Even though they all have these fancy names, if you really just approach the words by breaking them up, for example, up here, we see lipopolysaccharide. So we can divide that up into lipo, meaning fat, poly, meaning many, and saccharide, meaning sugar. When you have many sugars together, that's going to help you build a bigger carbohydrate. So again, all of these different smaller pieces are going to help us understand how those organic molecules come together to contribute to the overall functional anatomy of a bacterial cell. So remember, another goal of microscopy is to have that specimen contrast with the background. Sometimes that means that the specimen is dark on a bright background. Sometimes we're applying a stain to the background with a light specimen. And sometimes parts of the cell will have a different refractive index than other parts of the cell. So as long as the way that that material is bending light allows you to, or as long as that measurement of how it's bending light is different between different parts of the slide, different structures, then we will be able to bring contrast to that image. One more thing that you looked at last week is that, or I guess it's still technically week 1, but I'm filming this for week 2. So from the week 1 lectures, we saw that compound light microscopes are an example of bright field microscopy, that we have a dark image usually on a bright background, but sometimes we can stain the actual background and then have a bright sample on a darker stained background. So again, this is very common and routine. A lot of you have probably worked with light microscopes prior to going through this class. One other thing I wanted to remind you is that there is a microscopy worksheet. It corresponds directly with a couple of tables in your textbook, but I recommend not just copy and pasting what's in the tables, but really going through and filling those out as you listen to the lecture. Because again, sometimes the textbook is a very different level of detail than you need to know for the exams. And those worksheets are designed as study guides basically for the exams. The level of detail, you need to make sure you understand that from the slides. And then the review questions at the end of the worksheet are literally going to be questions that you see on quizzes and exams. So please take those seriously because they're really an important tool for making sure you're meeting those expectations for the exams. Okay, so now that we've kind of reviewed what we've already talked about and we have those thoughts in our head about the chemistry of life, we're going to think about how it comes together into definable structures. We're going to focus first on the difference between eukaryotes and prokaryotes, and then within prokaryotes, explore different functional structures. So in order to really understand the importance of what's different between prokaryotes and eukaryotes, you need to have a firm understanding of what is just common to every single cell. And the first is that it consists of a plasma membrane. That's not a completely solid structure. We classify it with the fluid mosaic model, so there's tons of embedded proteins and channels and receptors and all kinds of stuff just moving around constantly. But there is some delineation between the inside of the cell and its external environment. The inside of the cell is filled with a water-based cytoplasm that has a bunch of nutrients, proteins, electrolytes, and other things like that. So again, the goal here is to have a difference between the inside of the cell and the external environment. Sometimes the cell is in a condition where it's the same as the external environment, meaning it doesn't have to do anything to maintain its internal environment. But if, for example, the cell is in a hypertonic solution, like a salty solution, it's going to have to deal with constantly losing water from its cytoplasm to the surroundings. If it's in a hypotonic solution where it is salty compared to the dilute environment, it's constantly going to be taking on water. So I want you guys to keep that in mind, because when we get to bacterial growth and controls on growth, understanding some of those differences and adaptations will help you understand things like halophiles. All cells also have a nucleic acid blueprint, specifically in the form of DNA. Viruses have RNA in some cases, and there's different kind of forms of RNA and DNA that viruses have. But again, viruses are not cells, and their nucleic acids aren't organized into chromosomes. Regardless of whether it's a prokaryote or eukaryote, living cells have chromosomes at least some point in their cell cycle. They also have ribosomes. So ribosomes might be thought of as organelles. They're not membrane bound, so when we say that prokaryotes do not have membrane bound organelles, they still have ribosomes. And ribosomes are key for accomplishing protein synthesis. Proteins are the doers of the cell, and in order to have those proteins that makes a cell able to function and be unique, you have to have functional ribosomes. So these are things that all cells share in common. So just briefly, the differences between prokaryotic cells and eukaryotic cells. Usually prokaryotes do not have a nucleus. There's some really odd cases where they have kind of infolding that is similar to a nucleus. Generally, they just kind of keep their DNA in a specific location, which we'll get to in a moment. Eukaryotes do have a nucleus with a nuclear membrane, and some eukaryotes actually have multiple nuclei. With DNA organization, again, prokaryotes and eukaryotes both have chromosomes, but with prokaryotes, it's a circle-shaped chromosome with no special proteins that it's wrapped around. Eukaryotes do have those special proteins called histones, and they have linear chromosomes, so kind of shaped like a line. When we look at the difference in DNA replication and protein synthesis between prokaryotes and eukaryotes, that circle versus line is going to make a big difference. Prokaryotes generally don't have membrane-bound organelles, but life is weird and sometimes it happens. Eukaryotes generally do have membrane-bound organelles. Within the human body, certain organelles are found more often in certain types of cells and tissues, so not every eukaryotic cell has every organelle, but they generally at least have one type of membrane-bound organelle. Prokaryotes have complex peptidoglycan cell walls. Some eukaryotes also have cell walls. So for example, in fungi, the cell wall is made up of chitin. In plants and algae, remember algae are a type of protist, it's generally made up of cellulose and sometimes lignin. So a cell wall is just a support structure to protect the cell. It's found in a lot of different types of cells, but they're different materials. When prokaryotes and eukaryotes go through asexual reproduction, prokaryotes do that through binary fission, and eukaryotes do that through mitosis. So I mentioned that bacteria do not have a nuclear envelope, they don't have a nuclear membrane or a nucleus, but what they do have is a nucleoid. So this is not a membrane-bound organelle, it's just kind of a region of the cell where the bacteria keeps its DNA. So not constrained within a membrane, but still kind of organized using nucleoid-associated proteins, or NAPs. My class last semester really struggled with sleepiness because it's hard to go from an 8 a.m. lab into an 11 a.m. lecture, which we'll see once we go back into synchronous instruction. So I feel like we all can use a nap regardless of what our schedule is. So I like this slide because we all need some nucleoid-associated proteins. So when we're thinking about prokaryotic cells, so there's some different ways to classify them. When we know that we're working with prokaryotes, there's some really useful tools that we have, especially microscopically, for distinguishing between them. I showed you this on the first lecture, so looking at the shape of individual cells. When we talk about morphology, we're talking about individual cells. When we're talking about multiple cells and how they connect with one another, we're talking about arrangement. We can also look at the size of the cell. Some bacteria are radically bigger than others, and so that's going to affect staining characteristics. Really small bacteria don't stain well. It's going to affect virulence factors, and the way that we measure that is using a device called an ocular micrometer. That sounds super fancy, but ocular just means eye. Micro is like a microscope, something that looks at something tiny, and meter is a measurement. This is basically just a microscopic ruler that we pop into the eyepiece, the ocular lens. And some of you might have seen that in some of the microscopes if you came in person on Tuesday. Some of them had an ocular micrometer, and we can use that to actually measure the cells, but we have to calibrate it according to the magnification. This image is showing a protist called paramecium. That's not actually a bacterium, and you can see a bunch of membrane-bound organelles inside of it. So I mentioned that we can classify bacterial cells by their individual shape, their cell morphology. We can also classify them according to their arrangement. So here in this example, a coccyx is just a single coccyx, but if we have two of them stuck together, that's called diplococcus. If we have four of them arranged in a square, that's a tetrad. If we have a chain of cocci, that's streptococcus. And if we have a cluster of cocci that looks kind of like a bunch of grapes, that's staphylococcus. So you'll start to notice that some of these arrangements are also the names of different genera, which is the plural of genus. So sometimes we just name the bacterial genus or species according to its arrangement. You'll see Diplococcus is a genus, Streptococcus and Staphylococcus are common genera, Bacillus and Streptobacillus are also common genera. So Bacillus is just a single rod. Regardless of the genus name, if you see a rod-shaped bacterium, the morphology of that is Bacillus. If you see Bacillus italicized, that's referring to the taxonomic classification of that organism, not necessarily just the cell's shape. If we see a chain of rods, that's called Streptobacillus. So when we're thinking about why arrangement happens, why some cells remain stuck to each other in very specific shapes, here we see something similar to the previous slide. There's diplococci, streptococci, tetrads, sarcinae, which are these groups of 8, and then staphylococci. The reason for that is these are determined according to planes of division. So where the cell is actually going through binary fission, the plane along which that's happening might lead to an arrangement, might lead to some of the cells sticking together. So cell arrangement is determined by those planes of division through binary fission. When cocci divide to reproduce, and when bacilli do as well, some of those cells can remain attached to each other. They don't always, but if you have this single plane, like this sample A, you might get diplococci or streptococci. If you have these two planes that are kind of perpendicular to one another, you might get a tetrad. If you have this more complicated situation where you have I guess if we were in anatomy, we would call this transverse right here. And then we also have the sagittal and the frontal planes. But that's what leads to sarcinae. And then kind of random, not really organized division leads to staphylococci. But we can see that while all of these are circles, the way they look under a microscope is very different, especially with streptococci and staphylococci. There's a lot of tests that help us distinguish between those. So these are, for example, 5 different arrangements of cocci, but this happens to bacilli as well. So really the goal of this lecture, 20 minutes in, is to talk about staining and talk about how when we have different structures, different types of cellular anatomy, how we can stain them in order to visualize that. So some of the stains that you'll read about and try to observe with prepared specimens in the in-person lab is the endospore stain, which is pictured on the left, and the capsule stain, which is pictured on the right. Those are 2 stains that we're going to be exploring over the next couple of weeks. So like we talked about in lab yesterday, a sample has to be fixed and stained prior to examining it microscopically. When we say inoculation, that can be used as a kind of old-timey word for vaccines. Inoculation is introducing some bacterial or viral specimen, which might be alive, might be dead, might be able to cause disease, might not be able to cause disease, might start an immune response but not cause disease. You're introducing some form of microbial life. So here, inoculation is taking bacterial suspension from a broth culture or taking a loop full of water and then adding a little tiny chunk of solid inoculum from an agar plate. So you're taking some bacterial cells, you're putting them onto your slide so that you're able to dry them in place and manipulate them and stain and then visualize them. The drying can be done through air drying, especially if you don't have a lot of liquid. You can just leave it out and it will air dry. And then you can heat fix it by quickly passing it through the Bunsen burner just 2 or 3 times. Remember, when we're working with metal, you can absolutely stick it in the fire and let it get red hot and then cool down. When we're working with glass, like with a tube or with a slide, that is extremely dangerous. So whenever you're sterilizing glass, just quickly pass it through the same way that you probably did if you ever stuck your finger in a flame when you were little, how you just kind of pass it through quickly and it doesn't do any damage, but it does kill off any residual bacteria. So you do that. This process of drying and heat fixing is useful because it ensures that so there's a couple things to unpack here. The drying is important because if you skipped right to heat fixing, you would have liquid that would become steam, and that would destroy your sample. So you have to really thoroughly dry prior to transitioning into heat fixing. If you're using a slide warmer, it does that very gradually at the same time. So when it's set to a low setting, it allows it to evaporate, and then you're able to, at the same time, kill off those bacterial cells without damaging them, and then also make sure that they're stuck firmly into place. They're smeared and fixed onto the slide so that as you start pouring all this liquid on them, it's not going to do any damage. So that is accomplished either with a slide warmer or with the air drying and then passing it through the Bunsen burner. Okay, so getting into very specific structures, the first one we'll talk about in terms of functional anatomy are cell walls and cell membranes, and that is the basis of gram staining. So when we're thinking about which dyes to use in gram staining and other types of staining based on membranes, the charge of the stain really matters because that's going to affect how it interacts with those chemical structures of the walls and membranes. If we have a basic stain that is positively charged, most of your body material is negatively charged. So your membrane potential is often negative. You often have negatively charged DNA and proteins floating around inside of your cell. So opposites attract. You have to have this positively charged dye coming in and binding to the negatively charged cell structure. That's how we directly stain cells. If we want to do a negative stain, which is something that I'm going to demonstrate for you guys, I don't think you'll have time to do it in class tomorrow, but I will demonstrate it for you. This is where you are using an acidic stain that's negatively charged. Remember that our cells and bacterial cells are negatively charged, so it's like putting two negative magnets up against each other. They're going to repel one another. So you can do this without heat fixing and without changing the cell in any way. You basically add the dye and mix it directly in with a fresh sample and then drag out that stain and create a huge smear across the surface of the slide. You have to be careful because now those cells are still alive and they can cause disease if they're dangerous. But this is useful because, again, it's dyeing the background and not the cell. Doesn't shrink the cell size, so if you really need to measure the cell accurately, this is useful. It doesn't disrupt the arrangement in any way. Sometimes when we do the process of slide preparation, we do mess up the arrangement. That's why it's often hard to distinguish between Streptococcus and Staphylococcus under the microscope. And it can be really useful if heat fixing damages the cell membrane and wall too dramatically and makes it so that differential staining is not possible. So one goal of staining can be just to increase the contrast of what we're trying to look at. Cells naturally don't really have any color to them unless they're a cell that produces a special kind of pigment. So when we're looking at bacterial cells under a microscope, we do want to increase contrast, which is what we see with the simple stain of E. coli here. This E. coli has been stained only with crystal violet. But the E. coli on the right side, E. coli stands for Escherichia coli. It's a common bacillus, gram-negative organism that grows in our body and is found all over the place. So I say that it's gram-negative. We can tell that because of this pink color. This E. coli, the smear was prepared and heat fixed and everything. And then it went through a process where it was exposed to a few different stains. It was exposed to crystal violet and then Graham's iodine. That primary stain was washed off with either acid alcohol or acetone. And then a counter stain, safranin, was applied. So ultimately, E. coli only retains the safranin color. It loses the crystal violet. So the differential stain tells us how this E. coli is different from cells that do retain the crystal violet. So we'll kind of explore those differential stains over this section. When we're thinking about staining and why it's important to kind of be cautious about how we stain, one important thing to note is that we can damage the cell membrane and the structure after we go through the process of heat fixing. So in these images in the top right, the top left image within that collage is a bacillus that has been heat fixed, and you can tell it looks kind of fibery, kind of smooth. But then the one on the right has only gone through air drying and has been fixed with special chemicals. So you can see a lot of structural pieces there on the surface that you cannot see after heat fixing. So as we're going through differential slides, or differential staining protocols, it's important when you're heat fixing to be cautious about that, to recognize that sometimes the staining is going to be limited based on how badly damaging heat fixing is for the sample. So when we do kind of different types of simple staining and kind of unique staining like negative staining, that's used when we want to be cautious about size, morphology, and arrangement. And that means that heat fixing cannot be used. So when we're thinking about that special type of staining, which is sort of a simple stain, but sort of goes beyond that, we do that with a special protocol. So we put a little bit of the black dye onto a slide. We inoculate it with culture and we kind of move that around. And then we use a slide at a 30-degree angle to drag that smear across the slide. One thing to note is it's important to put the dye down first, because if you put the culture down first, that culture is not heat fixed, it's still alive, and there's always a chance that when you go to add the dye, as you drop it on, you suction back up some of the culture. So you just want to prevent that from happening by putting the dye down first. So then you let this dry, and by the time you're done, you're going to have a negative stain. The stain is going to stick to the slide rather than to the bacterial cells. You didn't heat fix it, so those bacterial cells are their natural size. You've gone through this special kind of different type of staining procedure in order to view these microbes. So when we're thinking about going through this process of staining in kind of complicated ways, especially if we're not just doing a simple stain or a negative stain, but really adding a series of stains, one really important thing to use is a mordant. This is something that really ensures that the primary stain, the first stain that we're using that will hopefully stick to unique structures, that primary stain is really driven into place and intensified by increasing its affinity, opening up the structure that it's binding to, or coating things like flagella. So the mordant in the case of gram staining is Graham's iodine. That's what actually encourages the crystal violet to really stick into place in the cell wall. I'm saying a lot of that verbally, but I'll have images for that in just a moment. In other cases, heat and fire are actually the mordant, so it can be a force rather than a chemical. That's what we do with endospore staining, which again we'll get to in just a moment. So when we're thinking about gram staining refers to the last name of a Danish bacteriologist named Hans Christian Gram. So gram staining and Graham's iodine are both named after him. So in the late 1800s, he was looking at lung tissue, so lung biopsies from different patients who had died of pneumonia. Pneumonia can be caused by many different things, and so he was trying to figure out, this was maybe like for them atypical pneumonia, not really sure what was causing it. He was just trying to increase the contrast between the microbes that were present and the lung tissue, but he noticed that when he tried this different staining protocol, some of the cells retained a purple color and others retained a pink color. So that tells us that there's something different between those cells. If they go through the exact same staining protocol and some of them hang on to one color based on chemical properties and others hang on to a different color based on chemical properties, there is a difference between them. So this is called a differential stain. It highlights differences between different organisms. And that is, again, based on structure, and it's our first kind of foray into diagnostics. So when we're thinking about the difference between a gram-positive cell and a gram-negative cell, gram-positive and gram-negative, first of all, only refers to bacteria. they have peptidoglycan cell walls. They're going to have those in almost all cases, regardless of, and a lot of other factors. So many cells will stain gram-positive or gram-negative. Some have different structures that get in the way of the stain, so they might structurally be similar to gram-positive, but they don't have a definitive gram staining characteristic. And then there's other branches of microbes as well, or different branches of bacteria as well. But gram-positive and gram-negative is a really great way to start to diagnose human pathogens. So again, both of them have a cell wall. Both of them have at least one cell membrane. In gram-positive bacteria, the cell wall's peptidoglycan layer is extremely thick, and they only have one cell membrane, the normal cell membrane that's found on all cells. In gram-negative bacteria, they do have peptidoglycan, but that peptidoglycan layer in the wall is extremely thin. They do have a second cell membrane that kind of compensates for that thin layer, and that second membrane has a lot of different interesting structural components built into it. So over here on the left, we see this teichoic acid kind of built into the peptidoglycan for gram-positive. On the right, we see a lipopolysaccharide. So we see this lipid A, we see this O antigen up here. That is unique to gram-negative bacteria, and it also causes a massive immune response in humans. So part of the issue with having a gram-negative infection is your own immune system getting too out of control. So when we see these differences, we can look at this artist diagram and say, oh yeah, the gram-positive looks very different from gram-negative and vice versa. But if you're just looking at a cell under a microscope, it's very hard to distinguish that. So going through, adding the different stains and relying on what you know about the chemistry and how those stains interact with gram-positive microbes, interact with the peptidoglycan, interact with the membranes, that's really going to allow you to make determinations about what type of cell you're looking at. Even though both of these have peptidoglycan, the structure of the peptidoglycan is a little bit different as well. So for gram-positive, there's a little bit more of this kind of lattice network. For gram-negative, it's a little bit more separated. That means that if something is a dye that's making its way into gram-positive and getting stuck there, there's a greater chance of it getting stuck versus in gram-negative, where that dye that would normally stain peptidoglycan falls right out. It's kind of like pore size. If you have kind of a thicker mesh, it's not going to let as much through, but if you have something like a volleyball net, that's going to let a lot of stuff through. So I'm going to kind of walk us through the gram stain process and explain how the dye kind of ends up interacting with gram-positive and gram-negative a little bit differently. I also want to try to do what I normally do on the whiteboard when I'm doing this lecture. So I'm going to have to redo it for each slide, but I think it's worth it. So this is the gram-positive. This is the gram-negative. With the gram-positive, we have a thin membrane down here. We have a membrane down here as well for gram-negative. And then for gram-positive, there's that thick peptidoglycan layer. For gram-negative, it's a very thin peptidoglycan layer. And then for gram-negative, they also have an outer membrane. I know that my lines are kind of wonky. I have to do all this annotation with my mouse, which is not very helpful. It's my birthday next month, so I'm going to ask my husband to get me a stylus. So hopefully after that, my images and diagramming will be a little bit better. Sorry about the delay there. So the first step here is to add crystal violet. So that crystal violet is the primary stain. It's going to really get into that thick mesh of the gram-positive, and it's going to stain the cells a very dark purple color, almost black. With the gram-negative, there might be some that gets into that wall, but remember there's an outer membrane here that might stop a lot of it. And even if it does get into that peptidoglycan cell wall, the pores are so open that it's just going to fall right back out. So in order to make sure that the crystal violet gets stuck in the peptidoglycan in the gram-positive, I'm going to click to the next slide. It's going to introduce more of the gram stain process, but it'll clear my drawing, so I'll just have to quickly redraw it. The next step will be to add the mordant, grams iodine. I just want to remind you that at this point, all of the cells look purple, regardless of whether they are actually gram-positive or gram-negative. So then we're going to add iodine, iodine solution or grams iodine. This is really going to make sure that the dye is going to stick into the peptidoglycan. So I'm going to draw the positive and negative. Again, for the gram-positive, it's a thick wall. Gram-negative, it's a thin wall. And then another membrane. So we already have the crystal violet really in place. Here it might just kind of be sitting on top or loosely affiliated. And then we're going to add the grams iodine. That forms a complex with the crystal violet, and it's kind of like putting your hand into a cookie jar. Your hand might be able to fit in and come out okay, but once you grab onto the cookies, you can't pull it out. So the crystal violet and iodine can enter into the peptidoglycan lattice matrix okay, but once they stick to each other inside of it, they are too big to get pulled out. So that mordant is driving that dye into that structure that it's binding to. With the gram-negative, you're going to have a situation where you have the iodine sticking to it, but the openings are still big enough that it's still going to come right out the first second it gets, or the first chance it gets. So again, I'm going to go on to the next step, and it's going to clear the diagram. So the next step is decolorizing. So I'm going to draw what we had so far. The peptidoglycan wall with the iodine and the crystal violet. Thin peptidoglycan cell wall with the outer membrane. And more crystal violet and more iodine. Okay, so another thing to note is there might be some extra crystal violet right here that might just get washed off, but what we're going to have happen is we're going to flood this with alcohol. It could be alcohol, it could be an acid-alcohol mixture, or it could be acetone. I usually like referencing acetone because I think a lot of people are familiar with that from clearing out Sharpie markings, or if you're removing nail polish, you know that acetone is a decolorizer. It removes dye. So for the gram-negative, where they have this dye just kind of sitting on the top, oops, wasn't supposed to get rid of the membrane, that's going to get removed and it's going to appear colorless. For the gram-positive, you might have a little bit of dye that gets removed, but the dye that got into that structure, the peptidoglycan, is really firmly in place and those cells should remain purple. So at this point, you can really identify just by looking at the slide. If it looks dark still, it's gram-positive. If there's no stain, it's gram-negative. But if you are in a situation where you need to look at the gram-negative, you still want to be able to have some level of contrast. So what you do there is you apply a counter stain. I'm not going to go ahead and sketch this out again, but the counter stain is a lighter color than the primary stain. It's saffronin. So if you've ever cooked with saffron before, you know it's kind of an orangey color. Saffronin is an orange-pink color. So for the gram-positive, this is kind of like drawing on a black Sharpie or black marker with a very light pink marker. It's not going to make a difference. It's still going to appear the color that it originally was. But for the gram-negative cells, if you add that pink color, it will make a difference. You'll be able to see that it's pink, and you'll be able to look at those under the microscope. Yesterday in lab, if you got a chance to look at E. coli, it should have appeared a pinkish color. That was through gram staining. So the positive here means that the bacteria is holding on to that primary stain. It's positive for the crystal violet Gram's iodine complex. Gram-negative means that the bacteria is getting rid of that primary stain. Once you go through the counter staining, it washes away that primary stain. So here we can see some kind of combination of the structure of the different bacterial cells, as well as the kind of insert into the cell and how it looks under the microscope. So some limitations of the gram stain technique, it's not perfect and it really does depend on your technique. So you can see here, there's a mixture here of the same shape of cell, but different colors. And that tells us usually that this was not a appropriately done gram stain. If there's a mixture, there's something wrong. So if you add too much acetone, this might actually be gram positive, but it's going to appear pink in some places because you got rid of that extra dye. So you stripped it of its crystal violet, and even though it should retain the crystal violet, you added too much acetone. If you don't add enough acetone, it might be the case that these are really gram-negative, but you didn't get rid of enough of the extra purple dye, so now it appears like a mixture. And if you use very old cultures, the cell walls start to weaken, even though it might look like an active culture, and even though you might be able to start a new culture with it, it's not good to look at old cultures under the microscope because their walls weaken, their membranes deteriorate, they don't interact with stains the same way as they did before, so it's not as useful. So gram staining is great, but like I mentioned earlier, it does not work on all bacteria. So mycobacteria, those include the causative agents of tuberculosis and leprosy. They have these waxy mycolic acids in their cell walls. That prevents them from retaining the stain, even though they would usually stain gram-positive. The mycolic acid causes it to not be able to bind to the peptidoglycan, and then you get a different result. Legionella and mycoplasma both cause pretty serious forms of pneumonia, and they don't gram stain. So it's really difficult to culture them in a lab, and it's hard to confirm that that's what you have. Really small bacteria, including mycoplasma, but others, and then intracellular pathogens like rickettsia and chlamydia, those are so small and in such a weird location that they don't pick up the stain very well. So again, gram staining is not the best diagnostic tool there. So gram stains are also differential, but they don't tell us anything about the differences within a single cell. You can distinguish between two different cells, but you can't tell anything about the unique structures within a single cell. That's why we need structural stains. So beyond the just simple distinction between gram-positive and gram-negative, we can look at those gram-positive organisms and start to account for those differences in their kind of cell wall composition. So acid-fast staining is specifically useful for identifying those mycobacterium that might cause tuberculosis or might cause leprosy, but that can't be stained according to gram staining. So this is kind of a structural stain in that it's picking up those mycolic acids. So we use carbofusion, which is a pink dye, and that is going to stick really nicely to those mycolic acids. We then use heat as a mordant, and we use acid alcohol to wash away the carbofusion from any cells that do not have those waxy mycolic acids. We counterstain with methylene blue, and then we're able to see pink where there's mycobacterium and blue where there's non-mycobacterial cells. So acid-fast staining is a slide that or stain that you'll hopefully be able to observe, and you're going to learn the kind of technique and general information for, but you won't actually do it. Okay, so if we were doing this live, I would encourage you to take a 2-minute break right now, but we are not. So if you're watching this recording, I encourage you to pause and give yourself a rest, and then get back into it. Okay, so getting really into specific functional anatomy beyond just mycolic acids and beyond just general differences in cell walls and cell membranes, endospores are a very specific structure. So again, those simple stains are useful for increasing contrast, but not great for diagnostics. Differential stains are a great first step for diagnostics. And then structural stains take it one step further. So here, this is Bacillus subtilis that has been stained using gram staining. It's gram positive, so it has this purple appearance. With the structural stain, we're going to use a dye called malachite green to highlight specific structures inside of single cells. So this gives us a greater level of detail about what this organism is doing, what kind of growth conditions this culture is in, and its ability to cause disease. So I wanted to give a quick example of Bacillus anthracis. This is the same genus as Bacillus subtilis, but you might be familiar with it because it's the causative agent of anthrax. It has a lot of features about it that allow it to be pretty dangerous. So it has that cell wall and plasma membrane to protect it. It's gram-positive, so just standard thick wall and one single membrane. But it also has its own DNA, and it has plasmids. Plasmids are small circles of DNA that allow you to code for superpowers, basically, in this case for different toxins. It also has endospores, which allow for propagation. Those kind of help enclose the plasmids and the nucleoid so that the genetic information is saved to survive through really dangerous situations. And then it has a capsule that prevents the host from basically capturing it with its immune system. It's like an invisibility cloak. So it has a lot of different virulence factors, things that increase its ability to cause disease. It can cause disease of the skin, which is cutaneous anthrax. It can cause respiratory disease, disease of the lungs. It can cause gastrointestinal anthrax, which is of the digestive tract. And it's usually associated with bioterrorism and terror attacks because some spores were sent through the mail to specific politicians and people. But it's actually most common in people who are routinely exposed to animal hides because the endospores actually end up in the dirt and they kind of stick around without a host. They get kind of onto the fur of different livestock like cattle as the cattle graze. And then as someone processes the hide, like if they're sorting wool or if they're making a drum, for example, then they would get exposed to those spores. Oh, I realize there's a typo here. This should not say Bacillus anthracis. I was trying to keep the formatting consistent. This should say Clostridium. So sorry about that, but this is another genus of bacteria that produces endospores. So there's Clostridium tetani, which causes tetanus. You might associate that with things like nails, rusty metal. The reason for that is endospores. Endospores survive and accumulate on these rusty objects. And then when those create puncture wounds, it creates an anaerobic environment that Clostridium loves. So that is why we associate those rusty nails with tetanus, because they help deliver the bacterium's endospores into an environment within us. That then causes what's actually called tetanus, the sustained contraction of the muscles, which is pictured here in this artwork of a soldier with tetanus, and it eventually causes death. It's very hard to treat, but it's very easy to prevent through vaccination. There's also Clostridium botulinum, which causes botulism. You might associate that with canned foods or with honey. This is why it's really important to not give honey to babies under the age of 1, because honey might have the Clostridium botulinum spores. So it does produce a toxin that can be deadly, but we actually also use the toxin for plastic surgery and different treatments. So Botox is actually derived from the toxin of Clostridium botulinum, which causes botulism. We can see right here, onabotulinum toxin A, that's the full name of Botox. So botulinum toxin A is referring specifically to the toxin it produces. So Botox can be used as aesthetically. It can also be used to treat a lot of pain conditions as well. So it has a lot of different applications, even though it's associated with something so deadly. So now that you've kind of seen some examples of where endospores are used, why do we use them? So bacteria really need to be able to reproduce rapidly. They make copies of themselves. They might be able to mutate and accumulate kind of different adaptations that way, but they do not get to engage in sexual reproduction. Sometimes they can swap different pili through conjugation, but they don't really have the routine form of genetic recombination that we have that allows us to adapt to different environments as a population. So what bacteria sometimes do is just wait it out. If they're not in a favorable condition, all they have to do is really save their DNA. They package it into this endospore, and then they're able to just wait until they're able to get into a situation with enough nutrients and then spring back to life. So some of these adverse conditions might include really high or low temperatures, low nutrients, extreme salt conditions, and extreme pH. So they can survive these really crazy conditions just by forming an endospore, a spore that forms inside the cell to protect the DNA. So endospores are a massive issue for space travel. It's really hard to ensure that all our equipment that goes out into outer space is free of endospores, because they can actually survive in space. So the Jet Propulsion Lab at NASA spends a lot of time really ensuring that they're able to sterilize their equipment from spores, because we don't want to introduce it to other planets, and then also ensuring or kind of observing how those spores change and survive and persist in outer space. So when we're thinking about how sporulation, the formation of endospores, happens, the DNA makes a copy of itself, or the cell makes a copy of its DNA, just like it normally would for binary fission. But instead of forming a whole separate cell, a special membrane just forms around that new pocket of DNA. So we see that happening right here. And that's going to be called the forespore. It's kind of like before you have the spore, this is the structure that is forming. And it's going to be building up membranes and layers until it kind of looks like this down here. So we eventually get this special protein coat around the cortex, and that's going to help break open the cell and release the spore. So we see this process right here. We see a cross section of the spore down here in the bottom left, and then in the bottom right, we see the distinction between the endospores and the vegetative cells that do not have endospores. In terms of the microscopy, we can actually kind of see this development occurring and the spore glowing. Different bacteria at different stages of development have the spore located in different areas. The location of the spore when it forms really helps us identify that particular pathogen. So, like I mentioned, a mordant is really important for ensuring that the primary stain sticks into place. Remember that endospores can survive outer space, they can survive extreme heat and crazy pHs. And your goal here is to break open that endospore so that you can stain it. That is a really lofty goal. And so what you end up having to do is basically apply steam heat to the bacteria while also trying to dye it for about 15 to 20 minutes. It takes a long time to really ensure that you've stained the endospore. Steam heat is very hot and it really breaks open that cell so that you can drive that primary stain into place. The primary stain is called malachite green. It's not actually from the malachite stone, but malachite stone is this really pretty green color and the dye is a similar color. So unless you have that consistent sustained heat while you're applying the primary stain, as you add the water to kind of rinse off the primary stain before you do the counter stain, it's just going to wash off. So the overall process here is apply that malachite green while also applying heat. Oftentimes this is done either with a hot plate with a beaker on it or a Bunsen burner with a beaker suspended over it. You kind of put a metal rod or a couple metal rods across the beaker to have a support structure for your slide. You cover your slide in a paper towel after the smear has been heat fixed and everything, and then you just basically let that water boil and release steam and just soak the paper towel in malachite green repeatedly. Whenever it gets dry, just add more, and that's going to help break open that endospore and get that primary stain in. Because it's so secure at that point, you can just decolorize with water. You don't have to use anything like a dye remover, and then you can just counter stain using safranin. So every part of the cell that does not have an endospore is going to be vegetative. It's going to turn pink, and any part of the cell that does have an endospore that's stained correctly is going to look green. So getting into some more functional anatomy, specifically capsules, there are a lot of different types of what are called glycocalyces. These are extracellular structures that kind of, so they're a bunch of different types of substance. They might be made up of sugars, they might be made up of proteins or both. Remember, polysaccharide means a complex sugar, like a carbohydrate. Polypeptide means a protein. So these are basically made inside of the cell and then pushed outward and accumulated on the outside of the cell. So they come from within so that they can be secreted out and form this protection. So again, there's different types of glycocalyces. There's a capsule, which is a very organized glycocalyx, which is firmly attached to the cell wall. There's a slime layer, which is unorganized and it's loosely attached to the cell wall. So they might be made of the same material, but their attachment in place is very different. There's also EPS, which stands for extracellular polymeric substances. That is secreted outward and it helps form biofilms to attach the bacteria to a hard substrate like your teeth or contact lenses or a catheter or a water pipe. So when looking at specifically the capsule for capsular staining, a capsule is an organized glycocalyx, again, that is firmly attached to the cell wall. So in this image over here, we see that capsule really held up nicely against the cell wall. That is going to be very important for keeping the bacterial safe from the host's immune system. These capsules are also non-ionic. They don't bind to any basic stains, and so if we want to be able to see them, we have to make sure basically everything other than those capsules is stained on the slide. So just to explain what we mean by aiding in bacterial virulence, virulence is the ability to cause disease, oftentimes by persisting and avoiding our immune system. So normally we would have antibodies that recognize the antigens on the bacterial cell. The capsule helps mask those antigens. So it's kind of like an invisibility cloak. Sometimes it can release protease to break down those antibodies, or it might just keep the cell hidden. So the capsules might break down those antibodies. That breaking down, or at least lack of interaction between antigen and antibody, leads to either blocked or just incomplete phagocytosis. So over here on the left, we see a couple of different types of what look like diplococci or diplobacilli. Some of them have a capsule, which appears like a halo. Some of them don't. The non-encapsulated bacteria are going to be more sensitive to the host immune system. So in order to visualize those capsules and get an image like we saw on the previous slide, we use something called albumin. Specifically, we use bovine serum albumin. Albumin is a plasma protein found in the serum of humans, chickens, cows. Sometimes it comes from eggs, which is white chickens. So a wide range of different animals. When it's specifically isolated from cow plasma, from bovine serum, it's called BSA, or bovine serum albumin. This is a standard protein that we use in a lot of lab protocols. It's also used to stabilize different reagents, different medications. So albumin is very sticky. It sticks to the slide, and when you flood the slide with dye, the albumin is basically going to force a negative stain. It's sticking to basically everything other than the cell, and that means that everything on the slide other than the capsule is going to pick up the stain, because the cell itself also picks up the stain. So ultimately we get something like this. All of this light pink in the background is where the albumin has picked up the stain. The dark pink in the center is where the cell has picked up the stain. And then the white halos are where there's a capsule that does not pick up the stain. So it's a forced negative stain done using carbofusion and albumin. So that carbofusion produces a reddish pink color that ranges in intensity depending on whether it's the albumin or the cell. The dye is going to stick to both the albumin and the bacterial cell, leaving only the capsule unstained, and again that capsule will appear as a halo. So some of these have much bigger capsules than others, some don't have capsules at all. Okay, so then I think this is the last bit of functional anatomy, looking at different structures that are involved in movement and causing a actual established infection. So there's a lot of different types of structures that are external to the cell wall. All of these cells, E. coli, Salmonella, and Shigella, are what we call enteric bacteria. They're gram-negative rods that live oftentimes in our gut. But if you look at them, some of them have these hairy structures. E. coli has a bunch of these longer structures coming out. Salmonella only has a few of them, and they're kind of coming out all over the cell, not just clustered in that one spot. And Shigella doesn't have any of those. It's just covered in that fuzzy stuff. So for bacteria, the benefit to having these external structures is to aid in motility and colonization of the host. For us, we take advantage of those by being able to differentiate between very structurally similar organisms. Again, E. coli, Salmonella, and Shigella are all gram-negative rods. They would all stain exactly the same and look exactly the same, But if we can visualize the external structures, then we're really able to distinguish between them. So some examples of these external structures include flagella, axial filaments, fimbriae, and pili. So we can detect whether motility is happening at all by growing bacteria inside of a special motility agar. This is called SIM medium. It detects the production of hydrogen sulfide, indole, and motility. We'll get more into those in week 3. If it's blurry like this, it means that the bacteria can move around. If the bacteria is all clustered in the middle, it means that it cannot move around. And we see that this one is non-motile. So flagella specifically are found in bacteria and some eukaryotes, although they differ in structure between the two. They're also found, a different version is found in archaea, and it's called archaela or archaela. So these are useful for moving along light gradients through phototaxis or chemical gradients through chemotaxis. There's also a special protein that's found on bacteria flagella called the H antigen. That helps us distinguish between very similar strains to really find out whether a pathogenic strain is present, something that can cause disease. So not just whether it can move, but also the arrangement of those flagella really gives us insight into what we're looking at. If the flagella is monotrichous, that means that there is only one single flagellum on one side of the cell. If it's amphitrichous, that means that there is a single flagellum on either side of the cell, each pole. Lophotrichous means that there's a cluster of flagella on one pole of the cell. And peritrichous means that those flagella are sticking out all over the place. They're covering the entire surface of the cell. So you can see here that flagella tend to be quite long, much longer than the actual length of the cell. So looking at how this appears, so actually the images here should be switched. This is what I get for trying to rush through this. Let me see, yeah, so this should go over here. This should go over here. I was trying to go from simple to more complex with the text, and then I just mixed up with the image. I'll fix the image before next week. Sorry about that. But this one over here is the polar, and then this over here with the bacteria all over the place is the peritrichous. Again, I'm so sorry about that. So when we're trying to visualize flagella under the microscope, it's a tricky process because we have these long projections that stick out from the cell. So you basically have to build up a layer of dye very gradually. We can just see whether there's movement at all using different types of augers. So there's MTM over here and SIM over here. We can also do flagellar staining. So again, here, different layers of dye were added sequentially in order to build up dye around the flagella. And it's a really delicate process. So flagella are kind of cute. They move in a couple different directions. So they move counterclockwise in order to allow the cell to move, but then they get kind of twisted up, so they have to unwind in a clockwise direction. And so that's what we see with this little cutie in the middle here. That is doing some tumbling. It's going through a clockwise rotation to unwind itself before it does a run like some of these other guys right here. Axial filaments you might have actually seen when you did the first lab, or if you looked at pictures of Treponema pallidum. Axial filaments are found in spirochetes or spirilla. These allow the bacteria to kind of move forward again, kind of like a corkscrew, but more of a constrained spiral motion with no runs and tumbles. This is really useful for these bacteria, not for us, but for the bacteria, because it helps them move through bodily fluids and then really embed themselves in the host. And then the last couple of external structures are fimbriae and pili. Fimbriae are a little bit shorter than pili, which is what you saw on the Shigella a few slides ago. Fimbriae and pili are both made of a protein called pilin. Oftentimes when you see IN, that means a protein, and these are usually found in gram-negative bacteria. Fimbriae, again, are shorter. They help attach to different mucous membranes like the intestines. That aids in the formation of aggregates, and they also help with forming biofilms. So again, really helping with virulence. Pili are a little bit longer. They might be involved in movement, so twitching and gliding, not runs and tumbles. There's also sex pili, which are involved in conjugation and direct DNA transfer between two different bacteria of the same generation. Okay, so that was the lecture for today. It's just about 1 hour and 15 minutes, so on track. Please make sure you're paying attention to those action items. The lab worksheets are not due until August 31st, but you should really try to be submitting them as you go along so that you don't have a bunch of them due all at once. There's also that microscopy worksheet I mentioned. Please, even though we're not meeting synchronously for lecture, please stay up to date with the lectures. Earlier today, I looked at the number of unique users that were viewing the lectures, and from the first day of class, only 10 people had actually looked at the slides. So I am begging you to please watch the lectures. It is so important for your success in the class.